Hi everybody! Alright, so here's part 2 of your notes for this unit. And in the first video we talked about DNA, the structure of DNA, and how it replicates or makes copies of itself. Now we're going to talk about how we use DNA to make proteins. Before that though, let's talk about the genetic code and how it's read in general. So DNA and RNA are read 3 bases at a time, or codons. So 3 bases code for 1 codon, and that 1 codon corresponds to 1 amino acid. Remember though, amino acids make up what important macromolecule? If you guessed proteins, you are correct. Alright, so we talked about DNA, now let's talk about RNA. So RNA stands for ribonucleic acid, and this time our sugar is ribose. It's still a nucleotide as a building block, so it's ribose as a sugar, a phosphate, and then bases. RNA, though, on the other hand, is single-stranded, so it's not double-stranded like DNA, meaning RNA can leave the nucleus, which is going to be pretty important in a little bit. The bases are also mostly the same, except for one, and that is uracil. So instead of thymine, we have uracil. Now this is where it also gets confusing, because even though we have uracil as a base, we're still making RNA from DNA. So DNA can still have thymine, in which case it would pair with adenine in RNA. Also, instead of just being one type of DNA, there's actually 3 types of RNA. There's M, or messenger RNA, R, or ribosomal RNA, and T, or transfer RNA. So mRNA is really important. That's what carries that initial code from the nucleus to the ribosome. rRNA kind of puts everything together, and then tRNA brings in that amino acid, which is important for making the protein. Here's just a side-by-side comparison of DNA and RNA. So you see DNA is over here on the right, RNA is on the left. DNA has C, G, A, and T, whereas RNA has C, G, A, and U, and then of course RNA is single-stranded and not double-stranded. Alright, so in terms of transcription, this is DNA making, my face blocks it out, but DNA making RNA. So this is going to occur in the nucleus because DNA can't leave the nucleus. So anything involving DNA has to take place in the nucleus. So what happens here is something called RNA polymerase, which is an enzyme, is kind of like an all-in-one. So it opens the DNA, it adds the free nucleotides, and ultimately creates that strand of mRNA. And the question is, why wouldn't you just use DNA? Well, like I mentioned, DNA can't leave the nucleus, but RNA can. So that's going to be really important for making proteins. So let's practice. We did this before with DNA, except this time, remember, we have a different base. So I'm going to do the first one with you together, and then I'll pause it and have you do the second one. So let me get a different color. All right, so A pairs with U, G, C, C, G. T pairs with A, C, G, C, G, G, C, A, U, T, A, G, C, C, G, A, U, T, A, U, C, G, T, A, G, C, C, G, C, G, A, U. All right, pause it here and try this on your own. All right, let's see if you got it correct. So GC, CG, G, A pairs with U, C, A, G pairs with C, then we have G, A, U, G, C, U, U, G, A, C, A, U, C pairs with G, T pairs with A. All right, so now that we've talked about transcription, let's get into translation. So we just made a molecule of mRNA, and now we're going to make a protein. So this occurs somewhere else. This time we're in the ribosome. And that mRNA, that's really important, brings codons. tRNA brings anticodons. And then it has an amino acid attached, which is going to continue to add to create a polypeptide chain, also known as a protein. And here's just an image showing that taking place. So we have tRNA comes in here, starts there, it moves to this site and this site, and you see the chain of amino acids forming. So here, to break it down a little more then, are your steps of translation. So first, the mRNA is going to attach to the ribosome. The tRNA brings your amino acid. The mRNA and the tRNA, they pair together, kind of like train cars on a train track. And then those amino acids are going to bond together, creating a protein. Pretty easy, right? Oh, but we got to talk about those sites. So within the ribosome, there are 3 tRNA binding sites, and this is the order they go in. So they start at A, then they go to P, then they go to E. So think E for exit, so E is last. P for polypeptide, where the chain grows. So A is where they enter, and they just have a single amino acid attached. And then P site is where it bumps the next one out, and it attaches to the growing chain. So this right here would detach when this one moves to the E site. And then when that tRNA gets to the E site, it's considered uncharged, meaning it doesn't have an amino acid attached. Now we need to talk a little bit about codons and translation. So codons are 3 base sequences that code for specific amino acids. And these are just the abbreviations you'll see in this table here. You'll see all the different abbreviations for the amino acids, but you read this table starting with the first letter, then the second letter, then the third letter. So for example, AUG is our start codon, and I'll kind of show you where that one is. So first, we start with A and U. So they all meet in this box. Well, then G, we go all the way across to here, and that's methionine, which is also a code for start codon. So methionine or start codon. And then you see there's 3 different stop codons. So now we're going to practice. So this time, before you do this, I would pull up an amino acid chart. That way you can practice doing this as we work through it. All right, so let's go ahead and translate this. So first of all, AUG, we know that that is the start codon. So start. According to my chart, CCG is proline or pro. UCU is ser. AAG is lysine or lyse, and then UGA is stop. All right, go ahead and pause and now. All right, so when you check your work for the second one, you'll see we do start, ALA, LYS, and then we reach stop. So that means don't keep going. You are just going to cross out whatever's left and stop there. Now, same thing here. We can't start until we get a start on the third one. So you can't do anything with CCC. Then you have start, GLU. Oh, we reached a stop. So we also can't do anything here. So now, unfortunately, things can go wrong. So mutations can happen, and mutations are permanent changes in a cell's DNA, but they don't always have drastic effects. Sometimes it's very minor, and sometimes it's silent or not noticeable at all. So these can occur both spontaneously or with chemicals and radiation. There are 2 main types. There's the gene and then the chromosomal. So the gene is going to be a single protein change, while the chromosomal is that entire chromosome changing. So when we review our cell types, somatic cells are body cells, and then sex cells are reproductive cells. So it's important to note that if there's a mutation in your body cell, it is not passed down. That's like saying if you have a scar on your body, your child is born with that scar, and that doesn't happen. Whereas mutations in sex cells are passed down. So when you have mutations there, if there's something wrong with the sperm or the egg cell, the resulting baby is going to have that mutation. When we talk about gene mutations then, there's several different types of point mutations or substitutions. There's a silent mutation, which is when the amino acid doesn't change at all. There's a missense mutation, where the amino acid just changes, but the sequence keeps going. And then there's a nonsense mutation where the change results in a stop codon. And if you look over here, here's an example of all those different point mutations. So you see the normal protein, silent mutation. You see, oh, we stopped early. This is incomplete, so it's a nonsense mutation. And then on this third one, you see a different protein. So that's how we know it's a missense mutation. There's also frame shift mutations that are a result of a substitution or a deletion, okay? Or not a substitution, sorry, insertion or deletion. So in this case, the base is either gained or lost. In a deletion, we lose a base. Just like you're deleting something, you lose a base. Insertion is the opposite. You add a base. So as a result, you look in this picture here, you see this was removed. So now the reading frame changes, and we also hit a stop way earlier than we should in making that protein. All right, so here's your final chance to practice. So you're going to practice with both the mRNA and then the amino acid chain, and it just kind of shows you how that has a difference. So when we do the mRNA here with the substitution, C pairs with G, C pairs with G, G pairs with C, T pairs with A, G with C, A with T, or sorry, not T, U, excuse me, and then C with G. So then when we have our substitution, we get G, A, C, U, G. So GCA is alanine, and then CUG is leucine. So now we have our replacement here. We have GAA. So GAA becomes GLU. Well, that's not alanine, so that's different, right? And then we still have CUG, which is LEU. All right, so now I want you to pause and do the other two on your own. All right, so go ahead and check your work, and you're going to see that there's some differences. So we started with ALA, THR. Well, then with that insertion of the base, it shifted our reading frame, and then we have ALA, TYR, and you can't do anything with that C. On the deletion, we have ALA and THR, but then on the deletion of the base, you have VAL, which is totally different, and you can't do anything with either one of those Cs. Chromosomal mutations then are the final thing, and this is in this change of the entire structure of the chromosome. So if you look at these different options here, you have deletion, where the B was deleted. You have duplication, where this B was duplicated again. You have inversion, where it starts like A, B, C, D, E, F, and then, oh look, D and E are gone, C, B are added onto the F as well. And then lastly, you have translocation, which is where it's kind of like crossing over, but where they switch and it causes a problem. So that problem it changes then is going to create lots of issues with the gene. All right, and that is it for your notes.